Good afternoon, children. Welcome to another session of biology class. This is my fourth video on our chapter, the circulatory system. In the previous video, I have explained the topic coagulation or blood clotting, and on this page I have written stepwise working, and I have already sent you. You should have written this in your classwork copies of biology. Today's topic is the most interesting in the whole chapter, and this is about blood transfusion and blood group. Let us start with the term transfusion. Sometimes it becomes necessary to inject blood into the body of patients that are undergoing surgical operation, and this process of transfusion, that is giving of blood to the patient, is called blood transfusion. Blood is taken from a healthy person, that is donor, and introduced through one of the patient's veins. But for doing this, it is necessary that the type of blood that is to be transfused should match, or it should be compatible with the type of blood of the recipient, that is the patient in this case. Alright, so this is what we are going to study today, right? Let's begin the process. Here, let's talk about the name, which is very important in the concept of blood grouping system. So, there was a person, Karl Landsteiner, and this concept of blood grouping was discovered by him. Alright. Now, to understand, suppose this is a blood vessel. Alright. This is a blood vessel. This yellowish color represents the base of the liquid matrix, that is the plasma. And I have only shown one of the blood cells, that is the RBCs. WBCs, platelets, I haven't shown. We are only concentrating on the process of blood group determination. So, RBCs of the human beings have specific proteins on the surface of RBCs, and we call them as antigens. Alright. On the other side, here the plasma of the blood has the complementary antibodies. Alright, once again, RBCs of humans, they've got specific proteins called antigens. Whereas in the plasma outside, right, they are complementary antibodies. Alright, there are two types of antigens on the surface of RBCs. The first type of antigen, or this protein, we call as antigen A, and the other type we call as antigen B. Now, depending on the presence or absence of the antigens, there are four types of blood groups. Suppose there is only one antigen present on the surface of RBC of the person, and it is antigen A. So, the blood group of that person would be blood group A. So, antigen is the determining factor, children, to determine what blood group does a person have. If it is antigen A, blood group will be A. If the presence is of antigen B, the blood group will be B. Now, sometimes both are present. So, if it is both present, it becomes AB blood group, and sometimes both are absent, right? So, in that case, when the antigens are absent, blood group becomes O. Actually, this O means 0, but it would look like very erratic or like very improperly given. AB, AB, 0. So, because 3 alphabets and 1 number will not make a right way of calling them, therefore 0 has been called as O. Alright. So, this ABO system, what we are studying is the presence or absence of antigen, and this is the main determining factor of blood group. Now, as I said, the complementary antibodies in the plasma, this will be just the opposite of what is the antigen. Suppose the antigen is A. Right? So, here only, what should be the opposite of A? B. So, the antibody would be B. If the antigen is B here, then the antibody would be A. Then, in the case when both the antigens are present, like A and B, then no antibody would be present. And finally, in a case when there are no antigens, that is blood group O, in that case, both the antibodies A and B would be present in the plasma outside. Also, children, make a note that the same way how it's in the textbook, the antigens, they are denoted by capital alphabet and the antibodies by a small alphabet. Alright. So this is how we determine the blood group of a person. I hope you have understood. This is very, very simple. The antigen on the RBC determines the blood group. And whatever is the antigen, exactly the same is the blood group. If it is A, blood group A, B, blood group B, both antigen A and B, the blood group will be AB. And if both are absent, the blood group will be O. O, as I told you, though we mean it 0, but we don't call it 0. On the other hand, the antibodies will be just the opposite of what is here. If this is A, here antibody in the plasma would be B. If this is B, then here the antibody would be A. If both are present as antigen, the antibody will be 0 or none of them. And if no antigens present, then both the antibodies A and B would be present. So I hope, children, this very simple explanation is clear to you all. We are studying the system of blood grouping, right? We are studying ABO system, right? Though there are several systems of blood grouping, but there are two systems which are most important. The first is, which we have studied like this, ABO, right? And the other is RH system, which we'll study just now. Now, let us talk about, according to this blood group system, which blood group can be given to a recipient, right? Now, we have understood that there are 4 blood groups in the ABO system: A, B, AB and O. And besides the transfusion of one's own type of blood, right, the other possible transfusions can be made in the possible ways like this. Let us understand here. This table, we also call it as the compatibility table. If you go left to right, that is if you are going horizontally, right, you will study the blood group which can be donated to which other recipient. And if you come down vertically, then you're talking about blood group that can be received. For example, A. The tick is on A and AB. What does this mean? That this A can be given to A and AB. But when you come down under A, then this means it can receive, this A can receive from A and O. Let us start, children. This is very simple and very interesting. The most luckiest blood group here, the people with blood group AB, they are the ones with the luckiest blood group because they can receive with any other blood group. For example, AB. In times of emergency, the blood group AB can accept from, see, look here. In times of emergency, how this complicated structure has come. This is what I am explaining. In times of emergency, AB can receive the blood from A, right? AB can receive the blood from B. AB can receive the blood group from its own donor like AB. Any donor of AB can give to AB, and also O. So, in the times of need, the people with blood group, they have no stress or no tension because anybody can give the blood to the recipient, right? For this reason, the blood group AB is called as the universal recipient. All right, that's what I said. They are the most lucky people, though they can receive from any other person as a donor. But now when you talk about the people with blood group O, they are the ones that are like social workers. They can give to all. Like O can give to A, O can give to B, O can give to AB, and O can give to O. So this is just the opposite. They are like social workers. They can give to all, right? And therefore we call them as universal donors. Once again, they are just the opposite as I said. AB, these are recipient. They can receive from all. We call them universal recipient. Whereas O, they are universal donors. They can give to all. So what comes about A and B? A can accept from O as well as from its own group, that is A. A can donate to A or O can give to A. Similarly, B can donate to B and O is already donating. So there are two people who can donate to B, that is B and O, right? The same thing is how this system has come, right? How about AB? AB can give to its own self, I've already told you, and all others are also giving. Similarly, when we talk about O, who can donate to O, children? Only O can receive from O. So this is like how it becomes in a way of like a social worker. It can give to all, but when it comes to receiving, it can only receive from O, right? So this is the compatibility table. Now let us see. A could give to A and AB, but while receiving, it could receive from A and O. B could give to B and AB, but while receiving, it could only receive from B and O. AB can donate to AB, right? It's a universal recipient, but when it comes to receiving, you can see it's receiving from all. And coming to O, right? O can donate to all, but can only receive from O. So this is what is the compatibility table. Coming to the RH system, I hope the ABO system is clear, children. It's a very, very simple thing to understand. What is antigen? What is antibody? How the blood group is determined? And why we call a particular blood group as A, B, AB or O? And how can we really donate or receive blood in times of surgeries or some accidents or some emergency? Alright. Now let's move on with the RH system. RH system, this abbreviation stands for, see, R capital H small. This abbreviation stands for rhesus. What is rhesus? This is our common monkey in which the factor was first discovered. So rhesus is the common monkey where the factor was first discovered. Now the blood of most people contains this factor. Most of the humans, they have this RH factor present, and when present, these people are called as RH positive. Now how we write RH positive? RH plus or RH positive, like this. Both are correct. So who's RH positive? A person who has RH factor present in his blood. Similarly, there are people who do not have this factor, and what do we call them? RH minus or RH negative. Both would be correct, and accordingly, RH negative is a person in whom the RH factor is absent. Alright. So when the blood of an individual, that is, let's say, RH positive, is transfused to a person that is recipient, and suppose he is RH negative. Try to understand this. Suppose this donor is positive. Just one second. Yes. Suppose this donor is RH positive and this recipient is RH negative. Now when the blood of this individual is transfused in a person that lacks this factor, then the person that is receiving develops an antibody for this RH factor that is coming as extra to its body. Once again, the person is negative and donor is positive. So the transfusion, immediately after the transfusion, the person who's a recipient develops antibodies, right? And these are for the RH substance that are coming from outside. In other ways, we call that this person gets sensitized. The recipient gets sensitized, right? So the first transfusion, the person who has received will not be dead, right? But he has got sensitized in about 2 weeks of transfusion. That is, within some 14 days, his body will develop antibodies against the RH factor. In other words, we call him that he's got sensitized. And the time period would be just 14 days. Alright. Now, suppose if a second transfusion is again given to this person, right? Suppose there's another transfusion. This is second transfusion given to this recipient, right? The antibody produced in his body already by first transfusion causes a reaction, right? With this antigen that is coming now and the antibody that has now been formed within 14 days, both will react, right? And this may even lead to death of the person who is receiving this. This is similar to development of an allergy, children. So the first RH positive developed antibodies, but if the second RH factor again comes here, then this RH factor and the antibodies, they will react, and this will be like an allergic reaction leading to death of the recipient. So this is what is the second transfusion. This produces a reaction like this. Antibody produced reacts with the transfused blood and causes the death of the recipient. And in a very rare case like this, suppose like this, Rh factor in pregnancy. Suppose the husband is Rh positive and the wife is Rh negative, right? The woman is Rh negative and the husband is Rh positive and they are married. Alright. Now the woman who is conceived with the child, and we are talking of first pregnancy, the woman is Rh negative. Now suppose this is the womb of the mother, is Rh negative, and the child, suppose by this genetic cross that we have already discussed in chapter 3, the child inside, suppose the child has the blood group of the father, Rh positive, mother negative, and child inside developing is Rh positive in the uterus. In this case, what will happen? The first Rh positive child will be normal, right? But if this sensitizes the mother, the second positive child, again if it is conceived soon, may have a problem, and this may lead to the death of the fetus and abortion. So such a marriage cross or a pregnancy in a mother with Rh positive blood group, Rh negative blood group, and the child being Rh positive because the husband is positive, so the first child would be normal, right? But if this sensitizes the mother and the second conceiving of or second pregnancy is again soon, then the second child may be dead, right, in the womb only, or it may get aborted. So this Rh factor is a very, very important factor. This must be very nicely matched during the transfusion, otherwise this will happen, right? This how we have examined over here. This can be asked in a number of ways in the exams, children, that what will happen if Rh positive blood is given to a patient or a recipient like this, right? You have to explain and you must know the reason why the death could be caused clearly. All right. So this completes the topic on blood transfusion and blood group determination and compatibility. I hope you have understood. The topic is very, very simple. You only have to understand these basic, very basic and easy concepts, right? The table, you should be very clear how you're moving across the table or down the table and what you're going to read with the stick or a cross. And then two systems, as I've already told you, ABO system and the RH system. Under RH system, there are two cases, that is how the transfusion could be a faulty one and what will happen and why. And similarly, how the Rh factor plays the role in pregnancy and why the first child is normal and the other child could be aborted. So that's all for just now, children. Next time, I'll be starting with my next video on the blood vessels. So read the topic. This is given in one page only in your textbook at page 93. And I hope you have understood. If any queries, please feel free to ask. Thank you.